events that are there. So first there's the development of endosperm, further by the development of embryo, ovules and ovary. The ovule part of the plant becomes the seed and the ovary part matures into the fruit. So always the ovule is going to be present inside the ovary. Now talking about the endosperm development, remember that endosperm is that nutritive part of the plant which provides nourishment to the developing embryo and thereby would provide the nourishment for the seed development and the whole entire development as well. Now you people remember that as a result of triple fusion, the primary endosperm nucleus is formed, right? Now this primary endosperm nucleus further it becomes the primary endosperm cell because it gets a cell wall surrounding it. It is still a triploid tissue only. Now, this primary endosperm cell, it divides repeatedly to form the endosperm tissue, which is again triploid. And since it is triploid, it has more, or you can say abundant of food reserve. And whenever the embryo will be growing, it will derive its nourishment, its proper nutrition from this endosperm tissue only. So now the primary endosperm nucleus, it undergoes two types of division. One that is free nuclear and the other that is cellular. So when it divides and the karyokinesis is not followed by the cytokinesis, it ends up having free nuclear endosperm. Later on, those free nuclei, they get a cell wall laid around them and that is called as the cellular endosperm. One such example which has the both, that is coconut, right? The coconut water is the free nuclear endosperm that it contains and the white portion that is eaten, that is the cellular endosperm present in it. Now, furthermore, in seeds such as the pea, groundnut, now see the endosperm could either be persisting in the seed, it could be completely consumed in the seed, or the remnants of the nucellus can be found in the seed. So all those seeds that have the persistent this thing, endosperm, right? We call them as the endospermic seeds or endospermous seed. And like basically the seeds that do not have it, we call it as non-endospermic seeds. Further, there is the embryo development. Now, embryo develops at the micropylar region of the embryo sac where the zygote was present. So, embryo is a result of the syngamete, not the triple, right? Now, the zygote, after it has been developed, it will start to undergo mitotic division, right? So as to form the new plants or the new plant products and stuff. So the early embryo development is called as embryogen. Now, this embryo development furthers certain stages like pro-embryo, globular embryo, heart-shaped embryo, and then the mature embryo. So, during the development of the embryo, these all stages take place and it is alike for the monocots and dicots. But the kind of embryo that is developed is different, obviously. You people know that dicots, they have two cotyledons present in them, right? So they have an embryonic axis and two cotyledons. The region above the embryonic axis is what we call as the epicotyle. The region below the embryonic axis is called as the hypocotyle. Now, epicotyle is the portion that gives rise to the plumule. And plumule gives rise to what, students? What does the plumule give rise to? No one.
What's the question?
The question is, plumule gives rise to what?
Shoot.
Shoot of the future plant is formed by the plumule. And then the region below the embryonic axis is what we call as the hypocotyle, which gives rise to the radicle, which is responsible for the formation of root system of the plant. Clear? Now, the same exact thing is going to be there in a monocot seed as well, a monocot embryo as well, but the little bit changes are going to be there because it is a monocot. So, it will just have a single cotyledon, which will be laterally placed And the name of that is Q-telem. In the monocots, there is also going to be radicle and plumule, but the only difference is that in them, the radicle and plumule, they are covered by extra layer of sheath to protect them. The plumule is covered by coleoptile and radicle is covered by coleorhiza. And root cap is present in both of these plants, whether it is dicot, whether it is monocot, root cap is present. Then talking about the seed, which becomes as a result of ovule being fertilized, right? So when the ovule becomes fertilized, or you can say a mature ovule forms the seed. Depending upon the kind of endosperm it has, the seeds are of three types: endospermic, non-endospermic, perispermic. Endospermic, they are also called as albuminous seed. They have the endosperm persistent. It's not that their endosperm is consumed by the developing embryo. The species are wheat, maize, barley, coconut, castor, sunflower. All of these organisms or all of these plants, they have their the endosperm persistent in the seed. Then pea, bean, and groundnut, they do not. So we call them as the non-endospermic seed or the ex-albuminous seed. Later on, there is the perispermic seed, and these are the ones in which the nucellus is persistent. Nucellus is found, and this persistent nucellus is what we call as the perisperm. That is why we're calling these seeds as the perispermic seed. Example, black pepper, sugar beet, these are all the examples of perispermic seed. Further, the seed has a protective layer surrounding it, which is the modification or the hardening of the integuments, which were there in the embryo sac or the ovule, right? And the seed coat is very tough, it's very protective in nature, and it doesn't let the seed suffer harm. It has two layers, outer testa, inner tegument are the two layers surrounding that the seed coat is made up of. Then in the seed coat also the tiny hole remains, which is the micropyle initially, and that is from where the oxygen and water would enter and allow the seed to germinate. And inside the seed, obviously, there's going to be endosperm, there's going to be embryo. Now, the last few topics related to this chapter are dormancy and seed germination, which are exactly the opposites of each other. When we talk about dormancy, it is the state of inactivity of embryo. Right? When the embryo doesn't get the nourishment, doesn't get the water or moisture or oxygen or all that it requires in order to grow. So it'll go into the state of inactivity, which we call as dormancy. But when you provide it with the necessary moisture, water, oxygen, and everything, then it is going to give rise to a little plantlet or a little seedling, and that is called as seed germination. In the time of the dormancy, the moisture content also decreases. It's like a survival time period. It's like doing everything to survive. Then talking about the advantages of seed to the angiosperms. So seed has, you can say, better adaptive strategy because it has a seed coat and everything. Once it is spread to different locations, it, you can say, give rise to a new plant over there. Then it has enough food reserve in it so that it can survive on its own and develop the new seedling. The seed coat is there, which provides the protection. And ultimately, because it is a result of sexual reproduction, it has, you can say, both the characters of both the parents, and it is superior in terms of genetic recombination. And they're very important because this is how our agriculture is propagated, and they show two properties, this dehydration and dormancy, because of which seeds are the best thing in order to propagate a new plant life. Just like how the pollen had pollen viability, the seed also has a certain time period for which it is going to be effective, or it will have the power of germination. So oxalis, like the seeds of oxalis, they lose viability within few months. Lupine, it is the oldest seed that was excavated from Arctic tundra, and it germinated after 10,000 years of dormancy. Imagine, it stayed alive for that long. Date palm, that is Phoenix dactylifera, it was excavated at King Herod's palace, and this was viable for 2000 years. These examples are very crucial in order to, like you must remember them for your examination. Also, always remember this as a thumb rule. However many ovules are going to be there in the ovary, that many seeds are going to be there in the fruit. The ovule can never exceed, okay, like the ovules that are there, it can never, like the ovary can never exceed the number of ovules. Yeah. Now, orchids fruit contain thousands of tiny seeds, but there are so many fruits that have a single seed present. So it varies from species to species. Orobranchi and Striga, these are the parasitic species that have several tiny seeds present in them. Then, talking about the fruits. So the fruits, they are the ripened ovary, obviously. The wall of the ovary becomes the wall of the fruit, and we call that as pericarp. Now, when we talk about the fruit, they are of three types, depending upon from where they are originated. True fruit, false fruit, and parthenocarpic fruit. Now, true fruit, they develop from the ovary, obviously, and false fruit develops from the parts other than the ovary, right? It can develop from the thalamus, and it can develop from various other parts as well. So, mango is a true fruit, whereas apple, strawberry, cashew nut, these are all the false. And parthenocarpic is a unique phenomena wherein the fruit develops without the fertilization taking place. And this is done obviously artificially with the usage of certain plant hormones. And we call this method as parthenocarp. And obviously the fruits that are going to be formed, they are going to be seedless because it's not a result of sexual reproduction. So why would it have the seed present in it? Now the fruits, depending upon whether it has a lot of mesocarp or not, it can be fleshy or it can be dry. Now fleshy fruits such as mango, guava, oranges, and then dry fruits such as mustard, coconut, all of them are there. Then we have apomixis and polyembryony. Now, apomixis is the phenomena wherein the seed is produced without fertilization. There, in parthenocarpy, the fruit was being produced without fertilization, and here, the seed is going to be produced without fertilization. And this is commonly seen in grasses and the Asteraceae family. Now, apomixis, it is a form of asexual reproduction that sort of mimics sexual reproduction in terms of the product, but definitely not in terms of the process. Now, apomixis further is of various types, one of which is adventive apomixis. Now, this is the kind wherein the embryo is developing from nucellus or integument, which is a normal diploid tissue, which is not responsible for developing the seed or the embryo. This takes place in the case of citrus, opuntia, mango. Clear? Lastly, we have the polyembryony, which is the occurrence of more than one embryo in a seed. This is found in citrus. And then there's this phenomena of apomictic polyembryony, wherein many hybrid varieties, they are made, okay, by inducing this thing, formation of seed without fertilization, and then those seeds having multiple embryos in them. So this will be a unique procedure, and these will be hybrids and they can be used in the agricultural process. So the farmer or the people that are cultivating plants, they don't have to go through the whole entire, process of sowing the seed, watering them, waiting for weeks and weeks for that to grow. So their reliance on the, you can say, environmental cues or the abiotic factors would be very much less. Now see, can you people see these questions? Can you solve these questions? Hmm. One second. Yes, you can solve these questions and tell me, then we'll discuss these questions and we have more questions to solve from this chapter.
Please hold on.
Everyone's done till here? Done, students? Done, students? Yes or no?
Yes, ma'am.
Okay. So let us discuss. So if an angiosperm male plant is diploid, female plant is triploid. Sorry, tetraploid. Find out the ploidy of the endosperm. What would be the ploidy of the endosperm?
Pentaploid.
Why pentaploid? Hmm?
Because the endosperm would like have a fusion between two polar nuclei and one male.
Because the diploid male will produce haploid gametes, the tetraploid female will produce diploid gametes. Isn't it?
Yes.
Hmm. So then what will happen?
Um, One sperm will fertilize to an egg and it will
Hmm.
form 3N zygote.
Hmm.
And then another sperm will fuse with two polar nuclei.
Hmm.
So that will also be 2N. So 2N plus 2N plus N will give 5N.
Very good. Exactly. Okay, when a diploid female plant is crossed with a tetraploid male, then what will be the ploidy of the endosperm? What did you people write for this?
Tetraploid.
Tetraploid. And then the last question, what will be the number of chromosome of alluron cells of a plant with 42 chromosomes in the root tip cells? How did you calculate this, students? Hmm? What did you do in this? Nothing. You didn't see the this thing also, right? The options.
Can you explain this?
Hmm. See, you people know that the number of cells or the number of chromosome in the alluron cells are found in the endosperm area. So that is 3N. Okay. Because you know that they are the result of your triple fusion. Now, the root tip cells, they will be diploid, isn't it? So the how are we going to calculate the whole entire thing? Because we know that the number of chromosomes, if we have taken any, n number, we have taken 42 chromosomes in totality, right? So when we talk about they're asking for the root tip cells, right? A plant with 42 chromosomes, and what would be the number of chromosomes of alluron cells? And we know that alluron cells are 3N. And what is N here? 21, because that is the number of chromosomes. So you multiply 3 with 21 and you get 60. Clear? Understood, everyone? Like they have given you the number of chromosomes in totality, right? 42. Half of 42 is the haploid number. And when you multiply that with the triploid 3N, you're going to get the number of chromosomes of the triploid cell. Is it clear to everyone?
Yes, ma'am.
Okay, very good. Sure, is it clear to every single student? Is there any doubt in the question that has been asked? Like these kind of questions, it may come in your exam. So you must pay attention. If you didn't understand, you tell me. I'll like, explain it again. Okay, everyone has understood. All right, so now today we'll start with the NCERT once again, the basic questions from NCERT, and then we'll move on to the further questions. One second, I'm sharing the screen. Okay, can you see the screen, students?
Yes, ma'am.
Yes,
Now
ma'am.
you can do the questions. Wherever you get stuck, ask me. Done, students? Done till here? Students, are you done till here? Done, students? Done till here?
Yes.
Done, students? Done till here?
Yes.
All right. Okay, so for today's class, we're going to keep it till here only and we'll continue in the next class. Okay, come prepared with this chapter. I'll take your Viva and we'll do more questions. Okay, that is
Okay, ma'am.
tomorrow only. Yes, okay.